Hello everyone, welcome to the class. So today we are going to solve the question paper. So that is a NEET test paper which is conducted on 22nd June 2022. Okay, so biology subject we are going to solve. Let us start solving, guys. Okay, so this is the first question. Okay, the question is identify the statement which is incorrect for Golgi apparatus. Okay, so in the options, what they are asking is, so you had to select the incorrect statement. Okay, you all know about the Golgi apparatus, right? So that is the organelle which is present in the cell, right? So this is from the chapter cell. Okay, so they're asking in the option one, they are telling that this phase of the Golgi apparatus is also called as maturing phase. Okay, so in the question, they're asking that incorrect statement, right? Obviously, the first one was only incorrect, right? The sixth phase, right? The sixth phase of the Golgi apparatus is called as a maturing phase. No, the sixth phase will be called as receiving phase. Okay, so it is not a maturing phase. So it is called as receiving phase. Okay, so it is called as receiving phase, right? So it is nearer to the endoplasmic reticulum, right? So that's why the protein which is secreted by the endoplasmic reticulum, the first it will come to the cis phase. Okay, so then the modification will happen in the Golgi apparatus, then it will go to the trans Golgi. Okay, that's why the cis phase we will call it as the receiving phase. Okay, not the maturing phase. Okay, then why these three options are wrong? Let us see. Okay, why these three options are wrong? So the Golgi cisternae are concentrically arranged near the nucleus. Yes. So these Golgi cisternae are concentrically arranged near the nucleus. Okay. So let us see the diagram. So this is the Golgi apparatus, right? So here you can see this is the cis phase. Okay. So here it was nearer to the endoplasmic reticulum. Okay. So our endoplasmic reticulum was nearer to the nucleus, right? Yes. So the protein which is synthesized in the endoplasmic reticulum, it will come to the cis phase. So then the modification will happen in this cisternae. So then it will go through this trans Golgi. Okay, so this we will call it as a trans Golgi. Okay, from the trans Golgi it will go. So here you can see that is the correct one, right? The yes. So here you can see the diagram of the cell. So here you can see the nucleus, and after the nucleus you can see the endoplasmic reticulum, rough endoplasmic reticulum. So then you can see the Golgi apparatus here, right? Yes. So it was concentrically arranged near the nucleus. So that is correct. So then coming to the option, the third option, Golgi apparatus pinch off from the tubules. Yes, guys. So here you can see the Golgi apparatus. So it is like a tubular structure, right? So whenever the protein was going to be transported, so it will be pinch off from the Golgi apparatus and it will be transported as a vesicle. Okay. So that's why this option is correct. The Golgi apparatus pinch off from the tubules. Here you can see. So this is the pinch off from the tubule. Okay. So this we will call it as a secretory vesicle. Okay. This secretory vesicles is having the proteins in it. Okay. Fine. Proteins and the enzymes on it. Okay. So that's why this third option is also correct. Okay. So then coming to the fourth option, some of the Golgian vacuoles function as a lysosome. Yes. So whenever the lysozyme enzymes, so which are the hydrolyses, are secreted from the endoplasmic reticulum, so it was transported from the Golgi apparatus. So in the Golgi, it will the bud off, right? So this vesicle it is formed, right? Some of these act as the lysozyme also. Okay. This bud offered vesicle is having the hydrolase enzyme which act as the lysozyme. Okay, actually our lysozyme was secreted or originated from the Golgi apparatus only. Okay, yes. So that is the reason this option 4 is also correct. So in the question they have asked that incorrect statement, right? So that is the reason the correct answer is option 1. Okay, option 1 is the correct answer, whereas the other options are wrong. Okay, because those are the correct for the Golgi apparatus. That's why. Let us move to the second question. Second question is the inner membrane of mitochondria. So you all know that powerhouse of the cell, right? Yes. So the inner membrane of the mitochondria. So they are telling that they are given the statement. Which statement is correct? Okay. So here the three statements are wrong. So here you have to select only one statement which is correct to the mitochondria. Okay. Let us go to the first option. Forms the infolding called as a thylakoids. Is that correct, guys? Thylakoid, where you have studied? Thylakoid we have studied in the chloroplast, right? So that is the reason option 1 is wrong. Okay, fine. So the thylakoid was present in the chloroplast, right? It is present in the chloroplast. We cannot see that in the mitochondria. So then coming to the third option, contain the 80% lipids and the 20% protein. No, guys. So here only so many people will get confusion. No. So here what happen in the mitochondria usually in some other membrane, some in the other membranes, 80% lipid will be there and the 20% protein will be there. Okay. So we'll all know that in the inner mitochondrial membrane, so there is more proteins are present. Okay. So that is the reason. So here you can see 80% will be protein. Okay, guys. So 80% will be proteins and 20% will be lipids here. Okay. So 20% we can see the lipids. Okay. So that is a speciality of the mitochondrial membrane. Okay. Fine. So let us go to the, so that's why this option is wrong. So then coming to the fourth option, form the infolding towards the perimitochondrial space. Okay, what do you mean by perimitochondrial space? We all know that the mitochondria is a double membrane, right? Which is the another organ having the double membrane. So that is a chloroplast, right? Yes. So what happened here? So this is the outer membrane, right? And here we can see the inner membrane, right? Yes. So this is the outer membrane, right? So this is the inner membrane, right? It is the inner membrane. So in between these two, there is a space, right? So that space we will call it as perimitochondrial space or intermembranous space, right? Two membrane are there. So that's why we'll call it as intermembranous space or the perimitochondrial space. Perimitochondrial space, right? So here in the diagram you can see, right? In the diagram you can see, so this is the outer membrane, this yellow color is the inner membrane, in between there is a space, so that is called as the perimitochondrial space. In the perimitochondrial space, there is no folding is there, right? So where the folding was happen? Inside the membrane, right? In the inner membrane, so there is a folding is there, right? Not in the perimitochondrial space. It is in the inner membrane, right? It is in the inner membrane. So that is the reason. Okay. So it is in the inner membrane. That folding, what we will call it as? That folding we will call it as cristae, right? Recall, guys, we will call it as cristae, right? So that's the reason option 4 is also wrong, right? Yes, option 4 is also wrong. What they've asked, which is the one correct statement, right? Correct statement they're asked, right? Yes. So that is the reason. So going to the second option, contain enzyme and electron carriers for the formation of the ATP, right? Yes. So in the inner mitochondrial membrane, our mitochondria is having the electron transport chain. In the electron transport chain, we can see the enzyme. So that is helpful in the formation of ATP, right? So we have a ATP synthetase enzyme, right, on the inner mitochondrial membrane. So that is the reason option 2 is the correct answer for this. Okay, guys. Option 2 is the correct answer, whereas the other options are wrong. Okay. Fine. So let us go to the third question. In the third question, they're asking, select the incorrect statement with respect to bacteria. So recall, guys, here they're asking incorrect statement, incorrect one. Okay. Incorrect means what? So 3 statements are correct here. 3 statements are correct, whereas the 1 minute, sorry. Yeah. 3 statements are correct, whereas the 1 is wrong, right? So 1 is wrong. So let us guess which is the wrong option. Okay, fine. So see here, let us go to the first statement. Heterotrophs are most abundant in the nature. So in the bacteria, we can see the autotrophs and the heterotrophs, right? Heterotrophs means which are dependent on the food for the other organism, right? Yes, so many bacteria are the heterotrophs, right? That is correct, right? Okay. So then coming to the second option, some members have the chlorophyll A similar to the plants. Yes, we have. Which is that bacteria, guys? Yes, cyanobacteria, right? Yes, cyanobacteria. Cyanobacteria, it is having the chlorophyll A and B. So that is that is an autotrophic bacteria, right? So sometime we will call it as a blue-green algae also, right? Yes. So that is the correct answer. So then coming to the third, some members play great role in recycling of the nutrients, right? Yes. Some of the bacteria which are helpful in the decomposition, right? So that statement is also correct, right, guys? Some members play a great role in the recycling of the nutrients. These are usually what? These are usually chemosynthetic. These are usually chemosynthetic autotrophs. Chemosynthetic autotrophs. Okay. So what they will do, while recycling the or while decomposing the material, so they will get the energy. So that they will utilize for the preparation of the own food. Okay. So that's why we are calling it as the chemosynthetic autotroph. Okay. Some bacteria are there, so that we will use it. So for example, you can take the Pseudomonas, right? Pseudomonas is helpful in the decomposition, right? Okay. So then coming to the fourth option, chemosynthetic nitrogen in the legume plants. Some of them will get the confusion here. So this is a wrong option, guys. This is a wrong option. Okay. So let us see what they're asking. Chemosynthetic autotrophs are involved in fixation of the nitrogen in the legume plant. You know what happened in the legume plant? We will call it as symbiotic association, right? Symbiotic association means here the bacteria is getting the benefit from the plant and the plant is getting the benefit from the bacteria. So it is fixing the nitrogen, right? So then what is the benefit that bacteria is getting? That is getting the carbohydrate, right? It is getting the food, right? So then how it can be autotrophs, right? It is not possible, right? Yes, it is a heterotrophs, right? So that's why the chemosynthetic autotrophs are involved in the fixation of nitrogen in the legume plants are not autotrophs. So they are heterotrophs, okay? So they are heterotrophs, okay? Because we know that they are having the symbiotic association, right? They are having the symbiotic association. They are having symbiotic association. So that's why they are the heterotrophs, not the autotrophs. Okay, guys. So that's the reason option 4 is a wrong. Okay. So they are asked that incorrect statement. So that's why option 4 is the correct answer. Okay. Fine. So let us eliminate the others. Okay, guys. Fine. So I think you understood this. Let us go to the next fourth question. Select the wrong match. Okay, what you have to do here is you have to select the which one is the wrong match. Okay, all the three are correct, but one is the wrong here. Okay, so 3 is correct. Okay, guys, 1 is wrong here. Okay, so let us find the which is the wrong one. Okay, fine. First one we will go to the Protonema, juvenile stage of the juvenile stage of the polytrichum. Okay, so polytrichum will belongs to the mosses. Okay, guys, mosses. It will belong to the mosses. Where you have learned the mosses? In the bryophytes. Okay, so if you take the bryophytes, in the bryophytes you have learned about the 1 minute. Yeah, so let us go to the bryophytes. Bryophytes. Okay, I will write here. Okay, okay. So let us go to the bryophytes. In the bryophytes, you have learned about the liverwort, right? Liverwort and mosses. Mosses, right? Yes. In the mosses, so you will see the protonema stage. Okay. So one we will see the primary protonema stage. Primary protonema is there. Primary protonema will form the filamentous-like structure. Okay. In the starting, it should be form some filamentous-like structure, right? That is the beginning stage. It will form the filamentous-like structure. So that's why they are telling that juvenile stage. Okay. So that is a primary protonema. Okay. So that will form the filamentous stage. And the secondary protonema stage will form the leaf structure. Okay. Secondary protonema will form the leaf-like structure. Okay. So then first one is a correct, right? Protonema we can see in the juvenile stage of the mosses, right? Yes. So that is the correct. So then coming to the second one, Markensia. So that is asexually reproduced by the gemmy. Okay. That is correct, guys. So Markensia, it belongs to the liverwort. Okay. So Markensia belongs to the liverwort. Liverwort. Okay. Because it is having the, so that name is a Hepatocopsida, right? Hepatocopsida. So that's why we will call it as liverwort. Okay. Hepato means what? Liver, right? Hepatocopsida. The example is the Markensia. So that will asexually reproduced by the gemmy. Okay. Yes. So in the Markensia, if you take the Markensia like this means on that there is a multi multicellular bud is there. Okay. So that we will call it as a Jama cup. Okay. So that we will call it as a Jama cup. Okay. On the Jama cup, so there is a Gemmy is present. Okay. So Gemmy is present. So that is helps in the asexual reproduction. So one Jama will, so there is a so many Gemmy will be formed. So in that singular one, we will call it as a Jama. Okay. So singular one, we will call it as a Jama. So this Jama will be detached. It will be detached from the parental, and so where it will go, so there it will be form the new plant. It will form new plant. New plant. Okay. Yes. So that is also correct. In the Markensia, we can see the asexually reproduced by the Gemma. So that is correct. So then go to the third one. Liverwort are having the unicellular rhizoids. Yes, guys. So that is also correct. So liverworts. So if you take the liverwort structure here, okay. So liverworts. So so liverworts does not have any true roots, true leaf, or the true stems. Okay. So they are having leaf-like, stem-like, root-like structure. Okay. So that root-like structure we will call it as a rhizoid. Okay. So it will be like this. Okay. So this is rhizoids, we will call it as rhizoids. So it will help in the attachment to the substratum. For example, it is a substratum. Okay. So it is a substratum. So it is helpful in the attachment to the substratum. Okay. Yes. So that is also correct. It is having the unicellular rhizoids. Okay. It is having unicellular rhizoids. Okay. That is also correct. So then coming to the fourth option, Funeria. Funeria is nothing but the bryophyte. Okay, that is the example for the bryophyte. Main plant body is the sporophyte. No, guys. It is main plant body is a gametophyte. Okay. The main plant body is gametophyte. Okay, not the sporophyte. It is a gametophyte. Okay. Gametophyte. Okay, gametophyte. So that is a haploid structure. 1N, right? Haploid structure. Okay. So haploid structure we can see only in the algae and the bryophytes. Okay. So remember always in algae and in the bryophytes. Okay. In algae and the bryophytes, we can see haploid structure. Okay. Haploid. The adult one is a haploid in structure. So that stage, predominant stage, we will call it as a gametophyte stage. Okay. Predominant stage, we will call it as gametophyte stage. Okay. So, but in the pterodophytes, in the gymnosperm, and in the angiosperm, we can see the sporophyte stage. Okay. 1 minute. Here I will write. Okay. Yeah. So then coming to the, yeah. So then coming to the coming to the pterodophytes. Okay. Coming to the pterodophytes, gymnosperm, and angiosperm. We are having the sporophytic stage. Okay, the adult one is in the sporophytic stage, so that is a diploid. Okay, so it is having sporophytic stage. Sporophytic, so that is a diploid. Okay, remember guys, so it is very important. Okay, fine. So I think you understood which is the correct answer, guys. Yes. So the correct answer is option 4, because they have asked here wrong match, right? It is not a sporophyte, it is a gametophyte. So other than options, others are correct. Okay. Let us go to the next question. Which one of the follow? Which one is not true for chlorophyce members? All you of know that, right? Algae. So chlorophyce is green algae, right? Chlorophyce is nothing but green algae, right, guys? Okay. So they're asking that which one is a not true. Okay, let us find which one is not true. The main photosynthetic pigments are chlorophyll A and chlorophyll B. Yes, chlorophyll A and B is the correct answer. Okay, fine. Then coming to the third, so produce zoospores with the 2 to 3 with the 2 to 8 flagella. Yes, guys. So it will produce the spores. So that is the asexual spores. Okay, that asexual spores is having the, these are the asexual spores. Okay, asexual spores. It is having the 2 flagella, 2 to 8 flagella. Okay, that spores is having the flagella. So that's why we'll call it as a zoospores. Okay, the flagella may be from 2 to 8 number. It will be vary. Okay, so that is correct answer. Store the material in the starch and the protein. Yes, so that is correct, guys. So it is having the proteinaceous structure, and where starch is forming the sheet-like structure here. Okay, yes. So it will store the food in the form of the starch and the protein. That is correct. So if it comes to the red algae, you know that in the red algae, Floridian starch will be stored, right? And if you come to the brown algae, so there is the mannitol and fucoxanthin will be stored, right? So that is the, sorry, mannitol and the laminarin will be stored in the brown algae, right? So like that. So here the starch and the protein will be stored. Okay. So that is the reason. So option 4 is correct and option 3 is also correct. Let us go to the option 2. Inner layer of the cell wall is composed of the pectin. So many of them will not notify this. Simply pectin is there. They will give an answer. No, guys. So whenever you are talking about the chlorophyll, here you can see it is having the two cell wall. Okay. So it is having the two cell wall. So here you can see this is the outer cell wall. Okay. The outer cell wall is made up of the pectin. Okay. So whereas inner cell wall is made up of the cellulose. Okay. Remember, guys. So the inner is cellulose. Okay. So whereas the outer is the pectin. Okay. So that's why option 2 is the correct answer. They're asking that not true, right? So they're asking not true or the false one, which is a false to the chlorophyll, right? The inner layer of the cell wall is made up of the cellulose, not the pectin. Okay. So that's the reason option 2 is the correct answer for this. Okay. And so whereas the other options are wrong. Okay. Fine. So let us go to the next. Transpiration pull. Okay, so what is transpiration pull? They're asking. So here you have to select the correct answer. Okay, it is positive hydrostatic pressure. Is that, guys? No. So here you have to remember. So which one is the main responsible for taking the water to the all the parts of the plant? So there are mainly two, right? One is the transpiration pull, right? So one is responsible is the transpiration pull transpiration pull, and the second one is a root pressure, right? Second one is root pressure, right? So and you have to remember that transpiration will be by pulling. Okay, pulling. Someone is taking the water from the plant, right? If you take the sun, if you take the whenever the temperature is more, on the time the transpiration will be more, right? The higher temperature will take the water from the plant, right? So that we will call it as a pulling. It is taking the, for example, I am sucking the water, right? If I am sucking the any fruit juice, so that is what we will call as pulling. Okay, I am taking the I am doing the pulling, right? So that we will call it as a pulling. Okay, transpiration pull, that is. Okay. So then coming to the root pressure is by pushing. Okay. So it is by, we will call it as pushing effect. Okay. So what will happen in the root pressure? So the water molecule, here the hydrostatic pressure will be like pushing. Okay. So it will push the water to the upward. Okay. So only in the alone in the root pressure, that it will not reaches to the all the parts of the plant. Okay. It requires the transpiration pull also for taking the water to the all the parts of the plant. Right. So transpiration pull is by pushing. Okay. So that is the reason. So here you can see root pressure. Root pressure will be the positive hydrostatic pressure, right? If I'm pushing means I require the energy, right? So it is by positive hydrostatic pressure. Here the water is putting the pressure. Okay. Positive hydrostatic pressure. Okay. So that is root pressure. Okay. So then coming to the transpiration is by the negative hydrostatic pressure, not the positive. Okay. It is by the negative hydrostatic pressure. Hydrostatic pressure. Okay, guys, it is by the negative hydrostatic pressure. So that is the reason option 1 is wrong. Okay. So it is not by the positive hydrostatic pressure. It is by the negative hydrostatic pressure because it is pulling, right? So that is the reason. Here the water is not putting any more of the effect. Okay, fine. So coming to the second option, provides pushing for the water transport. Is that, guys? I have told you that root pressure is for the pushing, right? Not the transpiration. Transpiration is pulling, right? It is taking, right? Pulling. So it is not like pushing, right? So that is the reason option 2 is also wrong. It will not provide the pushing. It will provide the pulling, right? It will provide pulling. Okay, fine. So let us go to the third. Result from the active absorption of water. Okay. So result from active absorption of water. No, guys, water will be from the diffusion, right? So the minerals will be taken by the active process, right? So the water will not take from the active process, right? It is by the diffusion. So water will be from, it will be from osmosis, right? Water will be always moves through the osmosis, not through the absorption, through the active absorption, right? So that is the reason option 3 is also wrong. Okay. So then coming to the fourth option, accounts majority of the water transport. Yes. So transpiration pool is helpful in the majority of the water transport. Yes. So that is correct statement. Okay. So that is correct statement. Okay. Fine, guys. Remember, so result from the active absorption of the water? No. So that is by the osmosis. Okay. So I think you understood this, guys. So the correct answer is option D. Sorry, option 4. Okay. Fine. So let us go to the next question. Enzymes of Tca cycle found attached to the inner mitochondrial membrane catalyze the conversion of. Here they are asking that you know about what is the TCA, right? TCA is nothing but tricarboxylic acid. Tricarboxylic acid pathway, right? TCA cycle is nothing but the tricarboxylic acid pathway. You know that. So this is the aerobic pathway, right? It is the aerobic pathway where the further breakdown of the our glucose will take place here, right? Fine. So the enzymes of the TCA cycle found attached to the inner membrane catalyze the conversion of, they are asking. Have you remember, guys? So there is a enzyme, right? So that enzyme name is succinate dehydrogenase. Okay. That enzyme name is succinate dehydrogenase. Here you can see the inner mitochondrial membrane. So here you can see there is an enzyme is okay. So this enzyme is helping in the conversion of succinic acid to the fumaric acid. Okay. So this is the mitochondrial membrane. Okay. So here the complex 1 is there. Here the complex 2 is there. So here, so this enzyme. Okay, 1 minute. In the last class also when I am solving the question I have told you. So here this enzyme is there. So that enzyme name is succinate dehydrogenase. Succinate dehydrogenase. Dehydrogenase, right? So where it is helping in the conversion of succinic acid to the fumarate, or we will call it as the fumaric acid. Fumarate. Okay. So that is the reason what happened here. So here you can see it will convert the 1 minute. Yeah, it will, where I can write. So here I will write. Okay. So it will convert the FAD to FADH2. FADH2. Right, guys? So then this FADH2 will enter into the ETC cycle, electron transport chain, right? So it is helpful in the ATP production, right? Yes. So that's the reason the correct answer is succinic acid, that is converted into the fumaric acid by the enzyme which is present on the mitochondrial inner membrane. So that is succinate dehydrogenase. Okay. Not the succinyl-CoA, not the alpha ketonic glutaric acid, so not the oxalosuccinic acid also. Okay. The correct answer is option 2. Okay. Whereas other options are wrong. Okay. So I think you understood this. Let us go to the next question. Complex 4 of the electron transport system is. Okay. So now only we have discussed about. So that is the electron transport chain which is present on the mitochondrial membrane. So they are asking that. So what is the complex number 4? Okay. So let us see. Complex number 4 of the electron transport system is cytochrome BC1 complex. No, guys. So it is not correct answer. Remember complex number 3. Okay. So complex number 3 name is cytochrome BC1 complex, right? Yes. So it is the complex number 3, right? It is complex number 3. So that is the reason option 1 is wrong. Then coming to the option 2, contain the cytochromes A3 and 2 copper center. Yes, guys. So when we are talking about the complex number 4, it is consisting of the cytochrome A and cytochrome 3. Sorry, cytochrome A and cytochrome A3, and it is having the copper ions also, Cu ions also it is having. Okay. So that is the reason option 2 is the correct answer. Let us see why the option 3 and 4 is not correct. Okay. Fine. See here option 3. What they are telling? Known as NADH dehydrogenase. No. Complex number 1 we will call it as NADH hydrogenase, right? Complex number 1. So then what we will call it as a complex number 4? Okay. So complex number 4, we will call it as cytochrome C oxidase, right? Cytochrome C oxidase. Cytochrome C oxidase, right? That's why not the third is not the correct answer. That is wrong. Okay. Then we are calling as fourth one is consisting of the F0 and the F1 part. No, it is not consisting of the F1 and F1 part. So it is present in the ATP synthase, right? It is present in the ATP synthase, right? So that's the reason it is also not the correct answer. Okay. Fine, guys. So the correct answer is option number 2. It contains the cytochrome A and A3 and it is having the 2 copper center. Okay, fine. So let us go to the 9th question. So it is from the genetics. So innate and biology. So the question is they are given the one pedigree. Okay. So here you have to select the which one is the correct answer. Okay. So whenever you see the pedigree, guys, you can tell that. So whether it is the dominant or the recessive one, right? Yes. So let us see here. So here in the option 1, they are telling that this pedigree is applicable to the hemophilia, right? Yes, it will applicable to the hemophilia. Here you can see, by observing this only you can see that. So here you can see the maximum affected is the males, right? So maximum affected when it is the male, we can see that it is the X-linked recessive disorders, right? Yes. So it is a X-linked, right? It is a X-linked recessive disorder, right? And it will come to the dominant also, right? It may be autosomal dominant also. We know that. So sometimes the autosomal dominant will be, you can see it will not skip the generation, right? It will not skip the generation. Here you can see, so in the first generation also parent is affected, whereas in the second generation is also affected, whereas in the third generation is also affected, right? We can also call it as a dominant, autosomal dominant disease also, right? So let us see one by one. So here select the correct option they are asking. Okay, fine. So let us come to the option 1. So given pedigree is applicable to the hemophilia. Yes, guys, it will applicable to the hemophilia because the maximum affected is the male here. So we can tell that it will call it as the hemophilia. You know that hemophilia we will also call it as the bleeder disease, right? Bleeder disease, right? Why we'll call it as a bleeder disease? So here, so there is a clotting factor 8, right? Clotting factor 8 deficiency is there, right? Clotting factor 8 deficiency is there. It will lead to the hemophilia, right? Fine. So then coming to this, so there is no clotting will be happen, right? So whenever the accident will happen on the time, so the blood will flow without the clotting, right? So that is due to the deficiency of the clotting factor 8. Okay. So there is no clotting will be happen here. No clotting, right? Okay, fine. Let us go to the second option. For a sex-linked character, genotype of the 1B is XCY. You know that XC means carrier, right? Okay. So let us see here. So if the male is there, okay, in the male, if it is a X-linked recessive disorder means, so there is no carrier, right? Only in the female carrier will be there, right? In the male, there is no carrier, right? Because only one X chromosome is there, right? So here it is not the XCY. Okay, this is the first 1B. What they are telling, 1B is the XCY. No, in the male there is no carrier. It is a normal, right? XY, right? So there is no carrier. So that is XY. So that is the reason option 2 is wrong, guys. Okay, I think you understood, right? There is no carrier in the male. Male will be affected, right? So option C is wrong. Okay, fine. Then coming to the option 3, this pedigree is not applicable for any autosomal dominant disorder. It will be applicable. Here you can see it is not skipping the any generation. Okay. So that is the reason it will be applicable to the autosomal dominant disorder also. Okay. That is wrong. Okay. So then coming to the option 4, for the sex-linked character, genotype of the 2B is XX. Okay. So there here you observe, guys. So what they are telling, in the sex-linked character genotype of the 2B. Okay, let us go to the second generation. So this is a 2 generation, right? Here they're asking the B. Okay, so B genotype is they're telling that XX. Okay, just observe here. If it is D, so father is affected here, right? Father is having the affected. Okay, so I will call it as the affected means we can call it as H. I will give H. Okay, So then Y. Okay. So then father is affected. So then coming to the XC, I will write. Okay. Father is affected and crossing with the mother. Okay. So crossing with the mother, who is carrier. Okay. She is a carrier. XC and X. Okay. So then what is the genotype of the girl baby, guys? XC, because from the father it is having the XC, right? It is having the disease, right? Diseased gene. So then the mother, from the mother, XC is another one is coming. So then it will be disease condition, right? It is a disease condition. So that's why here you can see the female is having the disease, right? So then it is not the XX, okay? It should be XX. Okay, it should be a diseased gene. Okay, so that is the reason option 4 is also wrong. It's not the XX. Okay, it should be a diseased gene. Okay, hemophilia gene. Okay, so that is the reason option 3 is wrong. So the correct answer is option 1. Okay, option 1 is the correct answer. Okay, so let us go to the next question. Find the wrong one for the catalytic converter. Okay, so catalytic converter. What is a catalytic converter? So in the car and in the some of the vehicles, we will fix this catalytic converter. Okay, so it is for reducing the pollution in the air. Okay, so what we will do? So we will fix this catalytic converter in the vehicles for reducing the pollution. So let us see which are the statements are correct for this. Okay, so it has costly metal like the palladium, platinum, and the rhodium. Yes, yes, guys. So here this is having the plating. So the plating will be there on the catalytic converter. The catalytic converter plating will be made up of platinum, palladium, and the rhodium. Okay, that is correct. So rhodium, it is the platings. Okay, platings. Platings is made up of platinum, palladium, and the rhodium. That is the correct answer. But here they're asking the wrong answer. Remember, guys. Okay. So go to the second question, second option. Convert the unburnt hydrocarbon into the carbon dioxide and the water. Yes. So what will happen? So in the so when these hydrocarbons will come into this catalytic converter, so it will convert this unburned this hydrocarbon into the carbon dioxide and the water. So then it will send it into the outer environment, right? So that's why it is the correct answer. Okay. Yes, it will convert the unburnt hydrocarbon into the carbon dioxide and the water. Correct. So then coming to the third option, automobile fitted with catalytic converter uses the leaded petrol. No, guys. So it is you should use the unleaded petrol. Okay. It should use unleaded petrol. Okay. It should use the unleaded petrol, because if we use the leaded petrol means it will spoil the catalytic converter. Okay. So it will spoil catalytic converter. Spoil converter. Okay. So that is the reason. So they will suggest that for which are the vehicles we are fixed the catalytic converter, they should not use the leaded petrol. So they should use unleaded petrol, leaded petrol. Okay. So this lead will spoil the converter. So that is the reason. Okay. So then it is a wrong option, right? Not a leaded petrol, unleaded petrol they have to use. So then coming to the fourth option, reduces the emission of carbon monoxide and the nitrogen dioxide and the hydrocarbon. Yes. So it will reduces the release of these harmful gases into the environment by converting it into the other gases, right? So they will convert this carbon monoxide into the carbon dioxide, and this will convert this nitrogen oxide into the N2. Okay. So this will convert the hydrocarbons into the water and carbon dioxide. Water and carbon dioxide, then they will release into the environment, right? So that is how this converter will work. Okay. So then the correct answer is option 3, because they have asked that wrong statement, right? So other statements are correct. Okay, fine. So let us go to the 11th question. Select the correct statement. Here we have to select the correct statement. Okay. Option A, they are telling area with the more species. For example, if you take one area is there, so there is a more species diversity is there. Different animals is there. Different organisms is there. It will show much variation in the total biomass year to year. Okay, so what they are telling that, so there will be a variation is present in the totally by year to year, there will be a variation will be present in the biomass. No, guys. So where, so there is a more diversity is there on there we can see that, so the variation will be very less by year to year. Okay, it is not the correct answer. The variation will be less. Okay, there is no much variation. The variation will be less year to year. Okay, fine. So then this option is wrong. Okay, coming to the option B, the stable community should show resistant to the disturbance. Yes, guys. So whenever you see the where in the biodiversity there is a more species is there and the stable environment is there, so there the disturbance will be very less. So we can see the tropical forest only, right? In the tropical forest there is the more diversity is there and a stable environment is there, right? So in that area we can see there is a less disturbance, right? Yes. So, and if you take the regions like the Antarctic regions and the Arctic region, so there's a more disturbances there, right? Because there is no stable communities also there, and there is species diversity also very less there, right? So that is the reason option B is the correct statement. Okay, yes. So coming to the third C option, as per revert Popper hypothesis, the rivet on the wings represent the key species. Yes, guys. So when we are talking about the revert Popper hypothesis, so it's like they have given this hypothesis like the aeroplane. Okay, so it's the aeroplane. So each nut is very important, right? So if we remove the one nut in the each time means it will lead to the aeroplane. It will not go in the stable condition, right? Yes. So like that, what will happen? So in the revert, the wings represent the key species. Okay. So as per the revert Popper hypothesis, the revert on the wings represent the key species, right? So whenever the key species becoming the it is becoming the less on the time, it will cause the instability in the environment. Okay, fine. So that is the reason option C is also correct answer. So the correct answer is option B and C. So that is the option 2. Okay, option 2 is correct answer. Whereas the option 1 and option 3 and option 4 are wrong statement. Wrong options. Okay, fine. So let us go to the next question. UTR right? Yes. So this is nothing but untranslated region. Okay. Untranslated region. Untranslated region, guys. Okay. So if you take the mRNA, okay. So if you take this is as the mRNA, 1 minute. Yeah. So if you take this is as the mRNA, so it is having the 5' end, this is the 3' end, right? Yes. So if you take this, the gene will start from here, okay? So the gene will start from here and it will end here, okay? So this is a start codon is here and the stop codon is here, okay? Stop codon is here, okay? So here you can see the UTR region, okay? On the 5' also you can see, and in the 3' also you can see the untranslated region, okay? So here also you can see, and in the 3' also you can see, okay? So this will helps in the translation efficiency, okay? So it is helps in the translation of the mRNA, okay? So let us see what they are given in the options. UTR. So present at the 5' end after the start codon. No, guys, here you can see. So here I have given the start codon, right? Here it is having the start codon. Okay. So they are telling that after the start codon. No, guys, it is present before the start codon, right? Before start codon. Before start codon, it is present, right? So that is a wrong option, right? So then coming to the option number 2, present at the 3' end before the stop codon. No. So stop codon is here, right? But it is present after the stop codon, right? So not the before. That is after the stop codon. After the stop codon, right? Before the start codon and after the stop codon, right? Yes. So here you can see the genes. Okay, fine. So that's why option 2 is also wrong. Some additional sequence that are translated into the proteins. Remember, guys, the name only telling that untranslated regions, right? So it will be not translated by the ribosomes. Okay, it is not going to be translated. Okay, so this is wrong, guys. Translated into the proteins. No, it is won't be translated. Okay, it is not translated. It is not translated. It is not going to be translated. Okay. So then coming to the fourth option, required for efficient translational process. Yes, guys. So it is required for the efficient translational process. For example, if you see, so it will be like, so from the starting it will happen. So from the starting it will just scan the thing, scan the mRNA. So then it will start from the start codon. So then it will exactly, it will synthesize the protein. So then it will stop there and it will continue that. Okay, but it will not continue the protein synthesis, right? So by holding and continuing the protein synthesis is required the untranslated region. Okay, so that is the reason option 4 is the correct answer. Okay, option 4 is correct. So whereas option 3 is the wrong. Okay, yes. So it is required for the efficient translational process. Okay, fine. Option 4 is the correct answer. So let us go to the 13th question. Read the following statement. Okay. So here they are given the three options. So three statement A, B, and C. You have to select which one is the correct. Okay. Some mammals have adapted to the fly. Okay. Some mammals are adapted to the flying. Yes. Bat is there, right? Yes. Bat is a mammal. Okay. Some are live in the water. Yes. Whale is there, right? Yes. Whale is there. Okay. So then this statement is correct, right? Some mammals have adapted to the flying and some are live in the water. That is correct. Then coming to the option 2, the long bones are hollow with the hair cavities in the bird. Yes. So in the bird we can see the pneumotaxic bone, right? Pneumotaxic bone. Pneumotaxic bone. It is a large bone. So that is having the hollow cavity in it. Okay. So it is a large bone. It is having the cavity in it. So that is for the hair space. Okay. So that is why we will call it as a pneumotaxic bone. Okay. So that is correct. So that is present in the bird. Okay, remember guys, pneumotaxic bone. Okay, fine. So then both the statement are correct. A and B is correct. Going to the third, tympanum represent ear in the reptiles and pisces. No guys. So you know what is the tympanum? Tympanum we will call it as the external ear. We will call it as external ear. Okay. So that is present in the reptiles. Okay. So that is present in the reptiles. Okay. Reptiles it is present. It is present in the amphibian and it is present in the mammals and it is present in the bird also. Okay. But it is not present in the fishes, spices. Okay. So that is the reason option 3 is wrong. It is not present in the spices. Okay. Yes. So then option C is wrong. So where the A and B is correct? Yes. So in the option 1, they are giving that option A and B are correct. Yes. Yes. So option A and B are correct here. So the other options are wrong. Okay, guys. Fine. So let us go to the next. Match the column with the column 2. Choose the correct answer. Here it is a multiple choice question. Sorry, it is a match the following question. Here you have to choose the which one is correct. Okay, fine. So let us go. So what they are telling here, cuboidal epithelium. So where you can see the cuboidal epithelium, guys. Cuboidal epithelium you can see in the tubular parts of the nephron. In the nephron, have you see when the urine formation is happening? So there will be a three process will be there, right? Absorption and there will be a secretion is there. So first will be the filtration, second one is the absorption, then third one will be the secretion, right? Yes. So where the secretion is happening, so there the cuboidal epithelium will be present. Okay, it will help in the secretion. So that is the reason. So for the A, 4 is correct. Okay. So then coming to the B, guys. Okay, all know that the brush border columnar epithelium, so that is helps in the absorption, right? So that is in the absorption. Then where the absorption will take place, so that is in the small intestine, right? Yes. So then it is present in the inner lining of the small intestine. So then for the B, 2 is correct, right? For the B, 2 is correct. Then coming to the ciliated epithelium, we all know that. So that is helps in the movement of the egg, right? So that is present in the inner lining of the oviduct, right? Yes. So that is present in the inner lining of the oviduct. So for the option C, third option is correct, right? So then we left with the fourth. So that is a squamous epithelium, right? So have you observed, guys, during the respiration, what will happen? So the carbon dioxide, right? So the carbon dioxide will be removed from the carbon dioxide will be taken up. So whereas the oxygen is diffused into the arteries, right? So that we have studied that. So that is helping in the so that is in the inner lining of the blood vessels. Okay. So that is squamous epithelium will be very thin. So that is helps in the absorption. Okay. So that is helps in the absorption. Okay. So that option D, the correct answer is 1. Okay. So that is the reason the correct answer is 4th. Okay. So I think you have seen that this squamous epithelium during the diffusion of the gases in the lungs, right? So near the lungs, you have seen that on the on the alveolar region also squamous epithelium will be there, right? Squamous epithelium is helping in the diffusion of the gases. Okay, fine. So that is also correct. So option 4 is the correct answer, whereas the others are wrong. Option 4, remember guys. Okay, fine. So let us go to the next. Read the statement and select the wrong one. So 3 statements are correct, 1 is wrong. You have to select the wrong one. In the prokaryotes, single RNA polymerase transcribed all the RNA. Which are those all RNA, guys? So RNA, which are those RNA? So there is a ribosomal RNA is there. So there is a mRNA is there, and there is a tRNA is there, right? Yes. So this all the RNA is translated by single polymerase. So that is in prokaryotes, that is in bacteria. Okay, fine. So that is correct statement, guys. Then coming to the option 2, in eukaryotes, RNA polymerase 3 forms snRNA. Yes, guys. So in the eukaryotes, what will happen? So there are the different RNAs are there. Different RNA polymerase are there. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay. So our RNA polymerase 2 will synthesize the mRNA 2. Okay. RNA polymerase 2. So then coming to this RNA polymerase 3, it will secrete. So it will synthesize tRNA and snRNA and another. So that is a ribosomal RNA. So that is 5S rRNA. Remember, guys. So all of them think that. So RNA polymerase 1, RNA pol 1 is there. So that will synthesize the ribosomal RNA, right? Yes, it will synthesize all the ribosomal RNA, but the 5S ribosomal RNA is synthesized by the RNA polymerase number 3. Okay, 3. Remember, guys. Okay. Fine. So this is also the correct statement. Then coming to the third option, sigma factor, right? The sigma factor acts as termination factor during the prokaryotic transcription. No, guys. So it is not acting as a termination factor. It is acting as initiation factor. Initiation factor, right? So that is the reason option 3rd is the correct statement, right? Because they're asking here wrong one, right? So that is the reason. So then coming to the 4th option, why it is correct. Okay. So let us see. Eukaryotes have the 3 types of RNA polymerase in the nucleus to transcribe the different types of RNA. Yes. So eukaryotes is having the 3 different types of the RNA polymerase. So that is also the correct statement. Okay. So that is the reason option 3 is the correct answer, whereas the others are the wrong. Okay, guys, option 3 is the correct answer. Let us go into the 16th question. How many molecules from the given below box having the molecular weight that is more than 800 Dalton? So what this is, we will call it as, guys, if it is more than the 800 Dalton means, we will call it as macromolecules. Remember, macromolecules, right? Macromolecules. So then coming to the here in the table, what they are given, adenine. Okay. So adenine is the unimolecular, right? So that is the micromolecule. 1 minute, guys. Yeah. It is micromolecule. It is not the macromolecule, right? So adenine, right? Okay, that is the micromolecule. So then coming to the lecithin. Lecithin is also micromolecule. Micromolecule lipids, right? It is the lecithin is a lipid. It is also the micromolecule only. Then coming to the DNA. No, guys. DNA is made up of the more nucleotides, right? It is the nucleotides, polynucleotide, right? It is the polynucleotide. It is not the micromolecule. It is a macromolecule. Okay, macromolecule, and it is having the more than 800 kilodalton. Okay. It is having more than 800 Dalton. Okay, fine. So then coming to the glycine, all of you know that glycine is amino acid, right? It is also the micromolecule. Then glucose, glucose is also the micromolecule. Then which is the correct answer? Yes, guys. So first option 1 is correct. There is only one molecule is there, so that is the DNA. Okay, so only one molecule is having the more than 800 Dalton. So others are wrong. Okay, fine. So let us go to the 17th question. Identify the incorrect statement with respect to the quasi-Urker. Okay, so you know that quasi-Urker in the last test we have discussed about the marasmus, right? So here quasi-Urker is there. So this is also the due to the protein deficiency. Protein, 1 minute, yeah, protein deficiency. It is also due to the protein deficiency. Okay, so in the option 1 they are telling that. So what is the question, guys? Identify the incorrect statement with respect to the quasi-Urker. Okay, incorrect statement. It is produced by protein deficiency accompanied by caloric deficiency. Okay, remember guys, kwashiorkor is not by the caloric deficiency. It is only by the protein deficiency. What will happen? So the mother will replace the mother milk by the any food which are more in the caloric food than the protein. Okay, so high calorie food will be there, but the protein deficiency is there in that food. Okay, so on that time the kid, so the infants will get the kwashiorkor disease. Okay, kwashiorkor is will be affect only for the children, so that is above 1 year age. Okay, so it will affect above 1 year age. Okay, above 1 year infants. Okay, so then it is not the calorific deficiency, it is only due to the protein. It is only due to the protein deficiency because high calorie food the mother is feeding. Okay, okay, so then coming to the option 2, it is occurs in the children more than 1 year age. Yes, it is occurring in the more than 1 year age. That is correct. So then coming to the option 3, it is shows wasting of the muscles and failure of the growth and the brain development. Yes, guys. So whenever there is a protein deficiency is there, it will lead to the it will lead to the protein deficiency and the growth will not happen. Okay. So and the brain development will also not happen. So that is the correct statement, right? So this is a correct statement. So then coming to the fourth one, extensive edema and swelling of the body parts are seen in the infants. Okay, so extensive edema and the swelling of the body parts are seen in the infants. What will happen? Here the kid, here the infant is feeding with the high calorie food, right? So what will happen here? So you can see the swelling and edema in the body part. Okay, so the swelling you can see in the body of the infant. So that is due to the more calorific food. Okay, so that is the reason option 4 is also correct. Okay, but in the marasmus we can't see. In the marasmus, edema and swelling we can't see. Or the body will be very thin on that time. Okay, fine. So that is the reason option 4 is also correct. And option 3 is also correct. Option 2 is also correct. The incorrect statement. So the correct option is option 1. Okay, guys. Because they're asking here incorrect statement. So that is the reason option 1 is correct answer. Okay. So this is incorrect to the kwashiorkor, whereas the other statements are correct. Okay, fine. So let us go to the 18 questions. Choose the mismatch. Okay, here we have to choose the mismatch. Cornea. That is the anterior portion of the external layer of the eye. Yes, so that is correct. It is the anterior, here you can see the anterior portion, that is the external layer of the eye, that is correct. So then coming to the fovea, the fovea centralis also we can call it as, right? So this is a thinning out portion of the retina where only cones are present. Yes. So in the fovea centralis, it is consisting of the high density of the cones. Okay. So where visual is maximum here. Okay. Vision is maximum here. High density of cones. Okay. High density of cones is there. And here vision is maximum. Vision is maximum here. Okay. Fine. So let us go to the third, retinal. Okay. So retinal, an aldehyde of the vitamins A. It is an aldehyde, right? Retinal is an aldehyde. So that is present in the vitamin A. That is correct. And they are telling that present only in rods. wrong, guys. Okay, so it is present in both rods and cones. Both rods and cones it is present. Okay, so the correct answer is here. They are asking that choose the mismatch, right? Yes, the third one is a mismatch here. Correct option is third one. Okay, so then coming to the why, fourth one is correct. Labyrinth, right? Labyrinth. So the labyrinth is the fluid filled inner ear, right? In the inner ear you can see the bony labyrinth and the membranous labyrinth, right? Yes, so that is a fluid filled inner ear. Okay, yes, so that is the correct answer. Here you can see the membranous labyrinth, right? Membranous labyrinth, you can see labyrinth, and bony labyrinth you can see bony labyrinth. Okay, so the second fourth one is also the correct answer. So that is why option 3 is the correct, so whereas the others are wrong. So the option 3 is the mismatch here. Okay, let us go to the 19th question. Select the incorrect statement with respect to the human skeleton. Okay, guys. So this is from the chapter Locomotion and the Movement. So here they are telling that sternum is a flat bone on the vertebral midline of the thorax. So what here you have to do? You have to do the incorrect statement here. Okay, incorrect statement you have to select. Okay, fine. So the sternum is a flat bone on the ventral midline of the thorax. No, guys. Okay, yes. So the sternum is the flat bone on the ventral midline of the thorax. Here you can see there is the ventral position. Here you can see the dorsal position. In the ventral you can see the sternum, right? So the sternum is there in the middle. Here you can see the rib cage, right? In the middle you can see the sternum. So rib cage you can see like this, right? Yes. So this is in the ventral position, right? So that is the correct. So the sternum is a flat bone. So that correct. So then coming to the second option, thoracic vertebrae, rib cage and the sternum together form the rib cage. Yes, guys. So how the rib cage will be formed? So rib cage will be like from the dorsal side, vertebrae is there, right? Thoracic vertebrae is there in the back side, and in the side there will be a rib cage will be there, right? So then ribs will be there, and in this and in the ventral side, so there will be a sternum will be there, right? So then it is formed the rib cage, right? Yes. So our rib cage is made up of in the dorsal side. Yes, in the dorsal side. So here we are having the thoracic vertebrae and the ribs on the side, right? Sternum will be on the ventral side. Ventral side. So this is my ventral side, right? So here you can see the sternum. So that is the reason option 2 is correct. Okay, fine. Each half of the pelvic girdle consisting of the coxal bone. Yes, guys. So where we can see the coxal bone? So that is in the pelvic girdle, right? So in the pelvic girdle, so we can see, so this side there is a coxal bone and this side you can see the coxal bone. Here the coxal bone is also made up of the 3 parts, right? It is also made up of the 3 parts, coxal bones, right? It is also made up of the 3 parts. So that is a coxal bone. So that is correct. Okay. So each one part, if I see this leg means here it is a one part. Here one part is made up of the coxal bone. And if I take the this side leg means here also one coxal bone is there. Okay. So that is also correct. Okay. So then coming to the fourth one. So the number of the girdle bone is equal to the number of the cranial bones. No, guys. So the girdle bone will be 6 in number. Okay. So girdle bone will be 6 in number, whereas our cranial bone will be 8 in number. Okay. So 8 in number. Okay. So that is the reason option 4 is wrong. Okay. So the girdle bone will be 6 and the cranial bone will be 8. Okay. So that is the reason the correct answer is here they are given the incorrect statement. The incorrect statement is fourth one. Okay. Fourth one is the incorrect statement. So that is the reason fourth one is the correct answer. So whereas the others are correct statement. Okay. Fine. So let us go to the next question. Yes. Read the following statement and choose the correct answer. Okay. The fluid filled synaptic cleft is present in the chemical synapse. Okay. So do you know what is the chemical synapse, right, guys? So here you can see the, so 1 minute. Yeah. So here you can see one of the neuron, right? Right. So this is the axon you can see. So here you can see the nodes, right? So it is a nodes of Ranvier. Here you can see it is the right dendrites. Right? So what will happen? So this is the presynaptic neuron, right? It is a presynaptic neuron. Presynaptic neuron is there. Here you can see the synaptic knob is there, right? So here you can see there is a membrane filled area. So that where, so the another neuron, right? So there is the another neuron. So that will connect to this, where the another neuron will connect to this, right? Yes, so it is a nodes of Ranvier, and here you can see the, so these are dendrites, right? Right? Yes. So here you can see, so there is a synaptic cleft is there, right? So here you can see what they are telling, that synaptic cleft, right? Here you can see this is a synaptic cleft. So this is a membrane filled space, okay? In this membrane filled space here, so the neurotransmitter will be released, okay? So in the chemical synapses, what happened? Neurotransmitter will be released here. Neurotransmitter release. Okay, neurotransmitter will be released here. So then it will be taken up by the postsynaptic neuron, right? So it is nothing but the postsynaptic neuron. Postsynaptic. 1 minute. Yeah. Postsynaptic neuron will be taken. So then the excitation will be happen here, right? So yes, the fluid filled synaptic cleft is present in the chemical synapse. So that is the correct statement. So then coming to the second option, all myelinated nerve fiber found in the central nervous system and the PNS are enveloped with the Schwann cell. All myelinated nerves found in the CNS and the peripheral nervous system are enveloped with the Schwann cells. No, guys. When we are talking about the central nervous system, so there are the two types of the cells which is required for the synthesis of the myelin sheath. Okay. So when we are talking about the central nervous system, CNS, so here the cells is required is oligodendrocytes. 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 It will secrete the myelin sheath. It will secrete the myelin sheath for the central nervous system neurons. Okay. The neurons which are present in the central nervous system. So the myelin sheath is prepared by the oligodendrocytes. Okay. So not the Schwann cells. Okay. So then we are talking about the PNS, peripheral nervous system. So here there is a Schwann cells will be there. Okay. Schwann cells will produce the myelin sheath around the neurons. Okay, it will produce the myelin sheath around the neurons, which is present in the peripheral nervous system. Okay, so what they are telling in the statement is the neurons which is present in both central nervous system and the peripheral nervous system. Schwann cells is only the one cell which will produce the myelin sheath. No, guys. So for the central nervous system, there is a separate cell is there. So that is the oligodendrocyte. For the peripheral nervous system, there is a separate cells is there. So that is the Schwann cell. Okay. So that is going to produce the myelin sheath. So that is the thing. Option B is wrong here. Okay. Statement B is wrong here. So then the correct answer is only statement A is correct. Okay. Yes. Only the statement A is correct here. Okay. So whereas only statement B is correct. No. Both the statements are incorrect. No. Both the statements are correct. No. Only option A is correct. Okay, guys. Fine. So then coming to the 21st question. So here what they are telling, 1000 primary spermatocytes and 4000 primary oocytes are there. Okay. So what it will form? Okay. So just remember, if the primary oocyte is there, okay, if the one primary oocyte is there means, so it will produce 4 spermatozoa. 1, 2, 3, 4. Okay. So it will produce 4 spermatozoa. Right. 4 spermatozoa it will produce. Okay. So coming to the 4 primary, sorry. So there are, if one primary oocyte is there, okay. If there is a one primary oocyte is there, so then it will produce only one egg. Okay. it will produce only one egg, or we will call it as a ova. Okay. So then here they are telling then, so 1000 into 4. Okay. 1000 into 4, it will lead to the 4000, right? Yes, 4000. So then there is a 4000 primary oocytes are there. Then what will happen here? The 4000 spermatid will be formed and the 4000 oocyte will be formed, right guys? So because 4 into 1000, right? 4000 spermatid will be formed. Spermatid, right? Spermatid is nothing but sperm. Okay. Then coming to the 4000 primary oocyte is there. Obviously, it will produce the 4000 oocyte only, right? Yes. So that is the reason they are telling that 1000 spermatid and the 4000 ova. No. 4000 spermatid and with 16,000 ova. No. 2000 spermatid and the 1000 ova. No. 4000 spermatid and the 4000 ova. Yes. So it will produce 4000 spermatid and the 4000 ova. Okay, guys. So the correct answer is 4000 spermatid and the 4000 ova. So that is the option 4 is correct. Have you remember, guys? So when the egg development is going on, so there is the primary oocyte will be formed and the, yeah, 1 minute. Yes. So primary oocyte will be formed. So that is a one egg only, right? So then the secondary oocyte will be formed. So that is also one only, right? From the secondary oocyte, the egg will be formed, right? Yes. So that is the reason. So the fourth option is correct. So then coming to the 22nd question. So I think so this is the last. If the alien DNA is ligated at the BAM-H1 site in the PBR322 vector, okay? So the transformant recombinant will be. So what they are doing here? So they are ligating the DNA in the BAM-H1 site. Okay. So what will happen here? They are telling. Remember, guys, in the BAM-H1 site, so there is a tetracycline gene is there, antibiotic resistant gene, right? So what will happen? Let us see now. Sensitivity to the ampicillin. Sensitive to the ampicillin. No. So in the vector, there is ampicillin gene is there, right? So there is no sensitivity. Resistant will be happen, right? So it is resistance. Resistance to the ampicillin. Okay, not the sensitivity. So then coming to the option 2, resistant to the tetracycline. No, guys. How the resistance is going to be happen? Because we have inserted the gene in the tetracycline, right? So now the tetracycline gene was damaged, right? It cannot produce the tetracycline, right? It will not produce the tetracycline because it was damaged by inserting the DNA, right? So that is the reason the resistance through the tetracycline is not possible. So what will happen here? So it will sensitive, right? Sensitive. Okay. So then in the option 3, they are telling that sensitive to the tetracycline. Yes, guys. So option 3rd is correct. Sensitive to the tetracycline because the gene has been damaged here, right? Because we inserted the gene in the tetracycline. So then coming to the resistant to the chloramphenicol. No, guys. So there is no chloramphenicol gene only not there. How can it will get the resistance to the chloramphenicol? Not possible, right? So that is the reason option D is also wrong. Okay. So the option 3 is correct statement. Okay. Sensitive to the tetracycline. Okay. Fine. So then coming to the last question. Which one of the following factor cannot activate the RAS mechanism? Okay. So what do you mean by RAS mechanism? Yeah, what do you mean by RAS mechanism? Where it will happen, guys? So RAS mechanism, have you this question is from the, yeah, this question is from the excretory system. Okay. This question is from the excretory system. RAS is nothing but renin, angiotensin, right? Then aldosterone mechanism. Okay, so that's why RAS. Okay, so renin, angiotensin, aldosterone, right? So that's why we'll call it as a RAS mechanism. What will happen? So the renin will be activated, right? So renin will be synthesized. So from the kidney. Okay, so renin will be synthesized from the kidney, and angiotensinogen will be synthesized from the liver. Okay, so renin will be from kidney. Okay, angiotensin will be from liver. Okay. So this, when the renin is activated, it is going to be activate the angiotensin 2. Angiotensinogen. Angiotensinogen. Then it is going to be activate the angiotensin. Angiotensin 2. Okay. So when this angiotensin 2 is activated, so then it is going to be activate the angiotensinogen 1. One minute. Yeah, 2. 2 is going to be activate the angiotensinogen 1. Angiotensinogen 1. Angiotensinogen 1. It is going to be activated into the angiotensin 1. Angiotensin 1. Okay. So when this angiotensin was activated, so it will activate the aldosterone. Aldosterone. Okay. So when the aldosterone was secreted, so it is helps in the increasing the blood pressure. Okay. So when it is going to be happen, guys, so whenever the blood pressure was very low, okay, so when the blood pressure was low, and so when the glomerular filtration was low on the time, so this angiotensin system, so that is the RAS mechanism, is going to be activated. Okay. So then the aldosterone is going to be secreted, so then it is helping in the absorption. Okay. Absorption of the minerals and the salts, so that is the sodium and the water. Okay. It is helps in the absorption of sodium and water in the, so that is in the nephron, right? Yes. So the option 1 is they are telling decrease in the body fluid volume, body volume. Yes, guys. So whenever the decrease in the body volume, so that is a decrease in the blood pressure. Okay. So when the blood pressure or the blood volume is decreases, on that time the RAS mechanism is going to be activated. Yes. So that is the correct reason. So then coming to the option 2, low glomerular pressure. Yes, guys. So now only I told you that whenever the low glomerular pressure is there, on that time also the RAS mechanism is going to be activated. That is also correct. Then coming to the low GFR, right? So that is nothing but low glomerular filtration rate, right? So low glomerular filtration rate. Okay. So when the glomerular filtration rate is low, on that time also it will lead to the absorption of the sodium and the water in the distal convoluted tubule. Okay. So it will help in the absorption in the distal convoluted tubule. Okay. In the DCT. Okay. In the DCT, so the sodium and the water absorption will be taken place there. Okay. Yes. So what do you mean by low osmolarity? Remember, guys. So when, for example, if you think that, so 1 liter water is there, so the salt concentration is very low, right? What we will call it as? So that is a diluted, right? So the diluted, so where the salt concentration is very less, right? So low osmolarity. Okay. So low osmolarity is nothing but high blood volume. Okay. So high blood volume. Okay. High blood volume. Okay. So whenever the high blood volume is there, on the time RAS mechanism is not going to be activated, because what is the function of RAS mechanism? So whenever the low blood pressure is there, on that time it will make that into the normal condition, right? So it will keep that into the normal condition. So that's why it is going to be activated. So here what happens? So in the low osmolarity condition, or when the high blood volume is there, or high blood pressure is there, so it will activate ANF. ANF. So this hormone is secreted from the heart. Okay. So ANF is going to be activated from the heart. Okay. So that is the reason option 3 is the correct answer. Okay. So whereas the other options are wrong. Okay, guys. So this is about today, guys. So if you have any doubts, you can ask me, you can ask us in the group. Okay. Fine. So that's it for today. Thank you so much. Let us meet in the solution of the next chapter test. Sorry, next NEET test. Okay. Thank you, guys.